Hello, my name is Kailee Henderson, and I'm from the SI Bridges program. With them this summer, we studied the spike protein sequences in the 5 U.S. states with the highest deaths for SARS-CoV-2. So what is SARS-CoV-2? This is a new strain of virus that's currently affecting the world. SARS-CoV-2 is the virus, and COVID-19 is the illness that it causes. SARS-CoV-2 is a mutation from SARS-CoV, and it attacks the respiratory system in humans. The SARS-CoV-2's virulence is very high because this protein is the S protein. Because of the way that this is shaped, it makes it very easy for the virus to enter the human cell. So it enters the human cell through a receptor, and the human cell attaches to the virus, then envelops it, and then moves it cell to cell in the human body, which helps it to attack multiple different organs. So as of July 10th, 2020, in the US, the top 5 states for highest deaths were New York, New Jersey, Massachusetts, California, and Illinois. So these are the deaths in those top 5 states as of July 10th, which equaled 140,120. So my research objectives were to compare the SARS-CoV-2 strains from the United States and Wuhan, China. Did those strains have a mutation? And if they did, where was it? And was the mutation correlated to the casualty ratio? So I started using bioinformatics as my main research method. I went on NCBI virus, where I focused my S protein sequences down to only 1,273 amino acids long, with completed sequences only from the United States and from China. Once that was focused, I downloaded 105 S protein sequences, gathered their FASTA files from NCBI, and then uploaded those to a phylogeny tree website called phylogeny.fr. From there, I took those files and renamed them on MegaX and then compared my data. So before I show you my tree, part of my results is the mutation. So we did have a mutation at the 614th amino acid, which is right here. So the mutation was from a D to a G. So the D mutation, the D strain, is the original strain that came from Wuhan, China, which somewhere along the way, until it got to the United States, it mutated and turned into a G. So D is the wild type and G is the mutant type. So here's my tree. As you can see, it splits in 2 different directions. Wuhan in California, a few from Illinois, Massachusetts, one from New Jersey, and a few from New York. They are my wild type, my D clade, the original that came from Wuhan, China. And then everything that branches off to the left being mostly Massachusetts, Illinois, New York, and then a few from California and one from New Jersey, or most of them from New Jersey down here. They are my mutant type. They are the G clade, the G variant in the mutation. So I think that when it mutated, it caused the casualty ratio to increase. So this supports that because California is only 1.93% for case fatality ratio, which is the original from Wuhan. And then all of the other states are at least 4% or higher, which is definitely an increase. So I believe that the mutation helped to increase the case fatality ratio. So using bioinformatics, we observed the S protein and its mutation from D to G. We observed the split in the tree from the D clade to the G clade. And then we observed the mutation from the S protein and figured that that is related to the high number of deaths in the United States. In the future, I would like to build a protein structure for the wild type and the mutant type and see how they differ for the spike protein. I would like to thank the SI Bridges program because they were an amazing help for this entire research. NIH for helping to fund my research, and especially Lakshmi, Jason, and Ellen. They were a big help. I wouldn't have been able to finish it without them, so they were really amazing. So thank you.